what's a microbiome? So microbiome basically is the community of the diverse microbes that reside on both on and within our body. So there are different kinds of microbes, like bacteria, archaea, fungi, protists, which reside in different body sites, and they play a very critical role in our health. Now, if you look into the microbiome in terms of pure numbers, so our body harbors around 100 trillion symbiotic microbes. Now, these microbes, like I said, they belong to different lineages, a wide variety of lineages ranging from bacteria, archaea, viruses, and protists. Around 95% of these bacteria, of these microbial groups, they reside in our gut, that is in our intestine. Now, if you look into the sheer numbers, the number of microbial cells that are on and within us, they outnumber our own cells by 30%. Like we have 1.3 times more microbial cells than human cells. If you look into the number of genes that they encode, or the diversity of the genes that they encode, it's even more. It's in fact 2 orders more than what we encode in our own genome. Now, if you look into these numbers, then we are a gigantic culture class of microbes. Like we are actually more microbial than actually human. Now, these microbial communities, they reside in different body sites, and depending on the body site, you have different compositions. Now, the microbes in different body sites are different. And not only are they different, they encode different kinds of functions, which enable them to both adapt to a given environment within our body, as well as to tune our physiology based on our requirements and their needs. And the most crucial of them is the gut microbiome. Now, the gut microbiome, as I mentioned some slides back, 90% of the human microbiome is next to the gut. And they have a diverse range of function. Like the most vital of them is the food, the digestion of the food. As I discussed, like they encode the number of genes that are encoded by the gut microbiome are more than 2 orders of magnitude higher than ours. So they play a very critical role in harvesting the nutrients from our food, breaking it down to nutrients that is absorbable by us, and at the same time providing nutrients that are essential for our well-being and for our survival. The second task they do is to regulate and prime our immune system. That is to give the immune system to tolerate certain groups of microbes, and at the same time prevent the colonization of different kinds of microbes which are called as pathogens or pathobionts. Now, the most important function that they also do is to produce secondary metabolites. Now, these secondary metabolites are metabolites like reputed, for example. Now, you might have, some of you who are in Twitter, they might have come across a recent story that there are some people who have this sudden urge to exercise. And this urge to exercise is dependent on the presence of a specific group of microbes which produce something called as fatty acid amines, which regulate our neural system, and they tend to have a more exercise or work harder. So there are these different kinds of stories, and one of the main functions of this is actually driven by the microbiome, that is the production of specific secondary metabolites. Now, because of this, as you can see, the gut microbiome, if you look into all the stories of the gut microbiome that have come up recently, you see more than 90% of the diseases that have been investigated have been observed to have some sort of association with the gut microbiome. Now, these can be diseases that are actually localized to the gut, like, for example, obesity, inflammatory bowel disease, insulin resistance, for example, or they can be diseases which are like arthritis, allergy, or eczema, which are a little distant. They are also been associated with diseases from the liver, like liver disease or cardiovascular disease. And not only that, the most interesting thing is they have been associated with even disorders of the brain, or disorders that control that are associated with our behavioral aspects, like appetite disorders, Parkinson's disease, Alzheimer's, etc. Now, what do we know about the gut microbiome? So this is from a recent review. This is from a review that is in the journal Gastroenterology. So what do you know about the gut microbiome? Now, there are two different aspects. We have our gut microbiome, and then we have the physiological aspect. We have the physiology, that is the status of our own health states. Now, there are different aspects in which these physiological, and there are different aspects which govern the host physiology. This can be aging, this can be diseases, And all these effects on the host physiology, they actually have an effect on the gut microbiome. This is one-way interaction. This is interaction type 1, where the gut microbiome actually senses something going on or going wrong with the host. And at the same time, so this is where it can come up as an efficient micro for diagnosis. That there is something going wrong and that the gut microbiome is actually sensing it. In a different manner, the gut microbiome, this change in the gut microbiome can have additional effects on the host itself, which is called the modulation power. So here the gut microbiome is not only simply sensing what is going on, it is actually causing something to actually either go right or go wrong in the host. However, the gut microbiome is not only directly influenced by the host, it is influenced by a range of other factors that are associated with the host, like the way we are, like our genetics, the way we were born, our lifestyle, like our lifestyle habits, like our sedentary lifestyle, or whether we consume alcohol or smoke, our gender, age, which is one of my key research areas. I will be giving 2 example studies on that. Diet, on which I will be talking about. Medications, which is another factor that is influencing the gut microbiome. And transmission. Transmission means from our parents, which is also driven by genetics, and at the same time from the people we stay with, or the people we meet, or the people we share our surroundings with. So this is basically transmission that we get from the outside world, the microbiome that we get from the outside world. Now, investigating these relationships is actually the focus of our area, which is called the data-driven, we can actually describe as the data-driven decoding of the gut microbiome, understanding the relationships. Now, how do we do that? Now, this is a generic workflow that is associated with any data-driven investigation. Like, we have a question or hypothesis, then we try to answer that hypothesis using some data-driven approach like machine learning, artificial intelligence, statistical learning, etc. And to answer that approach or answer that question, we have both publicly available data, and then we also can generate new data. And when we try to answer that question using specific data analysis approach, what we generally end up with is, we end up with answers or predictions, which we can again validate using further data analysis using additional data, or sometimes we get even more questions or more hypotheses, which starts a different kind of a analytical approach. So this is the overall analysis approach that we use in our area. Now, this is what we use, and I will be discussing just 2 aspects that I have worked on right now, that is called age and diet, and how they're related to the gut microbiome. So we'll start with aging. Now, aging is a multifactorial process. All of us will age, but we don't want to no one wants to age, but all of us will age, right? And here, one of the objectives that I have been pursuing right now is actually to identify microbial markers associated with aging and microbial markers that can actually distinguish between a healthy and an unhealthy aging trajectory. Now, one of the studies that recently came up was this review in Nature Reviews Gastroenterology, where we actually identified, when using a systematic review of more than 50 published aging microbiome studies, we identified specific markers or specific gut microbial markers, like you can see over here, that are associated with a healthy aging trajectory. If you look into this green and the gray group, there are different microbial markers that are actually associated with a healthy aging trajectory. When these markers go down, you have an unhealthy aging phenotype setting up. Now, this was a review that came up. So there were definitely different specific markers or different specific bacteria which are associated when an individual enters into an unhealthy aging phenotype. Now, next, I will also give you another example where we explore this further, using perhaps one of the largest gut microbiome meta-analysis involving more than 21,000 gut microbiome profiles from 30 different studies, where we actually identified specific bacteria within the gut microbiome which are associated with multiple disease or multiple disease phenotypes in both the older people as well as in the younger ones. And you can see over here, one particular example is for the healthy aging group, for the older individuals, where we see that there are clearly different kinds of microbiota which are showing a ranked order in terms of their association with unhealthy status. Like the ones in the bottom over here, they are generally showing association with the unhealthy status, and the ones on the top, they are showing association with the healthy phenotype. So that means that we definitely have markers We definitely have bacteria that we can target in the gut microbiome, but the thing is, how do we do that? Now, interestingly, this particular paper was recently highlighted by the MIT Technological Review in November 2022. And this question that is highlighted here, or they have already highlighted, is what I will be moving on to next. That is, from the perspective of diet, that is, can we actually tweak the microbiome to reset the microbiome checkpoints to regulate aging trajectory? So this is actually the topic of what I will be covering here in this, using one specific example study that I worked on. So basically, the gut microbiome, like Professor Barger mentioned, the gut microbiome as a connection between diet and health, or an example of how our gut microbiome can act as the transducer of the signals from diet of a beneficial diet to our health. Now, this story started off when I was in my postdoctoral research, and this is the story about a Mediterranean diet intervention called the New Age. Now, if you those of you who may not be aware, there are different kinds of dietary regimes that you might be seeing, like Paleolithic diet, keto diet, and one of these diets is called the Mediterranean diet. A diet that is actually associated with, traditionally associated with Europeans living on the Mediterranean coast, like Portuguese, Spanish, some Italian. So this is a kind of diet. And this type of diet, which I will be coming to next, is actually associated with prolonged health. It is negatively associated with cardiovascular disease. It is negatively associated with the onset of inflammation, inflammaging, inflammatory bowel disease, or even cancer. So it is associated with multiple benefits across the world. Now, what I will be showing here is an example, however, that dietary adherence to this diet was not exactly enough unless we had an intermediate gut microbiome response. Now, before that, I'll be just giving a detail of what a Mediterranean diet is. Now, if you look over here, Mediterranean diet is basically a diet which is rich in vegetables, legumes, fruits, nuts, olive oil, fish, and associated with a low consumption of red meat, dairy products, and saturated fats. And this is a very broad level thing, but they have individual classification schema or specific dietary guidelines that this should not exceed. Like vegetables, they should be higher this particular standard deviation. Red meat should not exceed this particular limit, and things like that. So they have strict dietary guidelines, but this is a very broad way of actually classifying what a Mediterranean diet is, and this is consistently associated positively with health. So in this intervention study, what we had two different cohorts from 5 different nationalities across 10 institutions, where we had a control group who were allowed to move on with their traditional industrialized dietary regime. There is the habitual diet, and then we had an intervention group who were given a specific Mediterranean diet. We were suggested a specific Mediterranean diet regime over a period of 1 year. And after 1 year, and both at the initial time point as well as after 1 year, we took observations pertaining to multiple data, like their cognitive function, how was it before the intervention, how was it after the intervention, their physical status, like their frailty, how frail, because these are elderly individuals. So how frail they were before the intervention and after the intervention, their inflammation status, their metabolome, like their blood metabolome profile, and finally their adherence, which we call as the Med-DAT adherence score. Now, once we did that, when we did actually, Now, once we did that, we first looked at the results, and we were not very happy. And if you look into all of these groups, between the interventions and the non-interventions, both in the beginning and the end of the like individuals who had an intervention, who took the intervention, and individuals who did not take the intervention, there was absolutely no difference in the dietary adherence scores, which meant that probably the diet didn't work out, and we were very disappointed. Then what I started to do was I decided to probe a little bit deeper. I asked, okay, overall there is no relationship, But do we see actually a relationship at the level of the microbiome? Now, when we started looking at the relationship, for example, now when we say that some microbiome or some aspect of the microbiome is probably associated with the diet, we can actually design machine learning models to predict the response, which is our adherence to the diet, from the microbiome profile. And once we suppose you want to associate A with B, what you can do is you can actually design data-driven machine learning models to see if you can predict from the microbiome the response to the beneficial diet. And what we found out was, yes, we could predict. Like you can see over here that with a reasonably sound accuracy, we could predict the dietary adherence from the microbiome profile, which was more or less positive, significantly positively correlated with the actual dietary profiles. And using something like a machine learning based, random forest based machine learning classifier, we could also identify specific bacterial species that were actually associated with this response. Now, when we looked into these bacterial species and we looked into their association with the host phenotype, we observed something very interesting. The green ones, the ones on the left over here, they are the diet positive guys. That is, they are responding positively to the med diet. And as you can see over here, if you look into their association pattern, they are actually positively associated with improved cognitive function, improved physical well-being, that is reduced frailty, and they are negatively associated with inflammation and increased frailty. If you look at the top guys over there, which are actually negatively associated with diet, that is, they go down when they're alive, they go down when we have higher adherence to diet, they show the opposite pattern. So it's an inverse correlation of it. So the diet response was correlated exactly the same manner with health as well respected of the med diet adherence, but it seemed to be driven by an intermediate microbiome response. Okay. Now then we looked into the functional aspects of these microbial species, and we identified that the species that were actually positively associated with diet, they were known to have functionalities or metabolites, they produced metabolites, they were known to produce metabolites that were generally associated with anti-inflammation, antioxidant activity, and better cognitive function. On the other hand, the bacteria that were, or the microbes that were negatively associated with diet were known to produce metabolites that were previously associated with colorectal cancer, cardiovascular diseases like trimethylamine N-oxide is a metabolite that is typically associated with cardiovascular diseases, Paracrisol, which is associated with kidney function, and then amides like putrescine, which are typically associated with inflammation. So what we are seeing over here is something very unique, like simple adherence to the diet, a healthy diet. It has no association with the overall change in the health. However, in those individuals who have a microbiome response, you see a clear association of, however, adherence to the diet has a significant association or a significant change with respect to certain microbial species. And when these microbial species or gut microbial species, they change, we have a significant response at the second level. So it's a stepwise phenomena which is happening. Diet adherence is associated with a microbiome response, and when the microbiome responds, you have a better clinical phenotype. So, and we then looked into the networks, like when you have microbiome, when you have a microbial community, you can actually build microbial association network, or interaction networks. And then what we realized that you can see the green ones over here, they are the guys who are associated with, positively associated with that adherence. And as you can see over here, the green guys are actually the center of the gut of this network. That is, these guys are key to holding the stability of the gut microbiome. On the other hand, the red ones are the ones on the periphery. So other examples, after our study, there are other examples which came up. So this particular study, it was done in US, and it came up in Nature Medicine, and they also identified that a simple med diet adherence was only associated with reduced cardiometabolic rate only if this particular bacteria was present in abundance. There was another study from the Queen CK cohort which were published in BMC Medicine, where they identified that intake of green degradation by default did not have any relation with inflammation, but only when the collegial abundance was there. This is another bacteria that these guys responded and had a beneficial response with respect to the inflammation. So, in summary, what this study shows is the gut microbiome is a sensor of the host health. It is also a modulator of the host health, but most importantly, it is the transmitter of bacterial signals on our health. So, but as you can see over here, there are several challenges, and which Professor Bell and some of which can be actually very efficiently addressed with some of the tools that Professor Bell's lab is currently developing. Now, one of the main area is the high interpersonal variations. Now, high interpersonal variations means that the microbiome that I have and the microbiome that someone else has, it might be very different. They're highly personal. So that means that we need better data-driven approaches for microbiome-based host stratification. We need to stratify the host so that we can suggest the appropriate dietary regime to them. Now, many of these cases, the food frequency questionnaires that are actually taken in to profile the diet are based on overall macronutrients or specific micronutrients. But something like many of the other aspects, like the flavors, for example, the chemicals, the spices, they are completely ignored, which are very key for determining which are especially the microbiome is very sensitive to these guys. So something like RecipeDB or FlavorDB or DietRx can actually be used to actually build this food frequency questionnaire at a much finer granularity. At the same time, many of the cases, in many of the dietary intervention studies, people generally, the data that the individuals provide is sometimes based on remembering. Like, you have to remember what you had 4 days earlier, or you have to just write it out. People can falsify that. But some tools like what dish, you can take the picture, you can see what you're eating, you can measure the calories, you can measure the content, and those will give you much better, tighter way of actually identifying the food frequency profiles of different individuals. So nevertheless, microbiome-targeted interventions are right now at a very initial stage. We will need much more efficient data-driven models to actually associate different kinds of, to actually stratify individuals based on multiple factors like our physiological status, age, our own habitual diet, our disease status, and at the same time, a combination of efficient diet measurement or diet profiling strategies with microbiome profiling strategies will enable us to come up with interventions or non-medication based interventions for improving human health in a much efficient manner. So with that, I would like to thank the organizers for giving me this opportunity to talk.